Humans driving another species out of existence is a story that we've heard time and time again. It's sad, but it's true. And we hear this story so often because we're in the middle of a mass extinction. It's the 6th mass extinction in the history of our planet, but it's happening orders of magnitude faster. The background rates of extinction are 1,000 to 10,000 times higher across all species than the previous 5 mass extinctions. And this one is happening so much faster because it's human-induced. It's driven by our activities, warming, polluting the planet, and our demand for natural resources. We've known this for a while, but what we still don't yet know is how much biodiversity we can lose before entire ecosystems become unstable and begin spiraling out of existence, bringing key natural resources with them. Natural resources that we use every day, whether we realize it or not. This mass extinction has everything to do with us and our survival. Traditionally, we've tried to combat extinction by creating protected areas, national parks, marine sanctuaries, and wildlife preserves that are designed to keep humans out, to keep human activity away, so that nature can recover and thrive. And we've been very good at doing this. We've been very successful at creating protected areas. In fact, this is a practice that has scaled exponentially. But biodiversity is still decreasing. We've lost 60% of animal populations since the 1970s, on average. This means that while some species are doing better than others, there are some that have been almost or entirely wiped out. Now, I'm not trying to say that national parks and protected areas aren't helping. I would really hate to see what these trends look like if we didn't have them. And we have had some really inspiring success stories about taking species and habitats that were on the brink of extinction back to healthy numbers. But what I am saying is that these efforts alone, they aren't enough to stop a mass extinction. Because protecting extremely vast, remote areas is incredibly challenging when the systems in place to protect them are far surpassed by the demand and the incentives to exploit them. Incentives that are amplified by a relative ease with which the natural resources taken from these areas can slip into the supply chain and provide huge payouts. But what if we can focus on the incentives? What if we can begin to understand and begin to work on the demand that's driving this trade rather than the symptoms? If we can work with human behavior rather than against it? These were questions that were first posed to me as a young graduate student by a nonprofit called Conservation X Labs. And they were stimulating and supporting new innovations that would replace unsustainable products. And they were beginning to develop new technologies that would replace unsustainable practices. And I became absolutely obsessed with these questions. And I really bought into this approach, and I changed my whole thesis around it. And I began working on timber and wildlife trafficking, which is a leading driver for extinction, and it's spurred by a poorly characterized demand for endangered species products. Now, normally when we think of wildlife trafficking, we think of poor creatures like this that are living animals that are transported for zoos or for pets. But timber from illegal logging is a huge part of this. And together, timber and wildlife trafficking is actually the 4th largest illicit economy in the world right now. It's just behind guns, drugs, and humans. But it's connected by the same international cartels that are well organized, highly funded, and much, much smarter than we care to admit. Like these other types of contraband, illegal timber and wildlife products are trafficked in a way that is designed to avoid detection. So these supply chains are poorly characterized and poorly understood. But unlike other types of contraband, we're faced with another challenge. And it's that while some products are very easy to identify, inspectors are on the lookout for pangolin and for pangolin scales. Although maybe not all of you know what this animal is, it's currently the most trafficked animal in the world right now. But almost all of us in this room, we can easily identify what an elephant tusk looks like on sight. But most illegal timber and wildlife products are just that. They're products. They've been processed into some form that makes them completely unrecognizable from their source. Are these steaks? Are these from a cow or a pig? Or are they from the pangolin that I showed on the previous slide? And these posts, they look like they're from a type of cedar, but are they from an eastern red cedar, or are they from an increasingly threatened Spanish cedar that requires CITES permits to trade? The fact that these illegal products can be so easily disguised as common things we use every day makes them incredibly difficult to detect. But one thing that doesn't change about them is their DNA. And scientists have known this for a while. Conservation biologists about 20 years ago at the University of Guelph developed a technique called DNA barcoding. And we've heard of barcodes, but in this case, DNA barcodes, they refer to a specific genetic sequence that is unique to individual species. And this technique of DNA barcoding has been very successful. It works very well at figuring out exactly what species an unknown sample or an unknown product came from. But this technique, it does require a highly trained scientist and a fully equipped molecular lab. These are resources, and this is time that inspectors just don't have. They're often faced with a 2 or a 3-hour window within which they have to make a decision on a shipment before it passes through and disappears into the supply chain. That simply isn't enough time to collect a sample, send it to a wildlife forensics lab to have it processed, and a result interpreted and returned to the inspector. The people on the front lines of illegal and timber wildlife trafficking, they need a new tool that can be used to identify an illicit shipment wherever they encounter it in the time frames that they have to work with. And this was a problem that I focused on with my doctoral work, through which I developed a proof of concept platform that I demonstrated using a microfluidic chip that performed a rapid DNA test. It was using a technique that was actually developed for disease diagnostics and global health. And this platform, now called the DNA barcode identification tool, or the DNA bit, is now being translated for commercialization by my team at Conservation X Labs. When launched, the DNA bit will accomplish 2 goals. First, it will serve as a screening tool that inspectors can use to screen products for illicit shipments, stopping them before they reach their intended markets and cutting off the payouts and the incentives for exploiting natural resources. And second, this platform is being designed to keep track of illegal and legitimate shipments in order to provide an unprecedented amount of information about this complex and deceptive supply chain. Information that we can use to understand where illegal products are coming from, and more importantly, where they're going to. Because if we can begin to understand what's driving the demand for them, then we stand a chance of developing sustainable replacements to take the pressure off of these precious ecosystems. However, in its current form, the DNA bit is really limited to a certain subset of sample types. Sample types like these steaks or fillets that are characterized by a large amount of readily accessible DNA that can be captured very easily. My work as a Schmidt Science Fellow has looked to really expand and push the envelope on the types of samples that can be addressed with rapid DNA screening. Sample types like timber that are characterized by an extremely small amount of DNA that's entrapped in really dense, tough tissue. Getting DNA out of timber samples is so difficult, even in the lab, and getting it out without breaking down what little DNA is left is so challenging that specialized tools are used that have to process this tissue under liquid nitrogen. These are methods that don't translate well for use in the field. When I was thinking about this problem, I became really curious if focused ultrasound could be used to disrupt tissue and accelerate and enable DNA extraction. And so I started my Schmidt Science Fellowship working at Virginia Tech with Dr. Eli Vlasavlovich, studying a specific type of focused ultrasound called histotripsy. Now, histotripsy was invented in Eli's PhD lab at the University of Michigan by a team of clinicians and scientists. And histotripsy, histo-tissue tripsy breakdown, was developed by Eli as a non-thermal, non-invasive therapy for liver cancer because of its ability to break down liver tumors. Now, this method works by having a scaffold that aligns ultrasound transducers that focus ultrasonic waves, very similarly to how a magnifying glass can focus light waves passing through it. However, instead of heating up that point, these convergent ultrasound waves, they create a point of really high negative pressure that's above the intrinsic threshold of water, meaning that it actually rips apart that surrounding media, creating a void space that will coalesce and grow into a dense bubble cloud. You can see the bubble cloud forming in the video. The lasers are just so that you can see it better, but this is created all with that convergence of ultrasound waves. In the collapse of these bubbles, the collapse of this dense bubble cloud creates really strong shear forces that can rip apart tissue. In the surgical suite, It's been used for its ability to collapse and liquefy solid tumors into an acellular debris. We've built and expanded upon this technique to create a method of focused ultrasound extraction that similarly is able to disintegrate tissue and accelerate DNA extraction. Using this technique, we've been able to show that we can get useful DNA out of it, DNA that can provide for a definitive species identification. And we also know that we are able to break down timber tissue. This is a sample of white pine that we're treating with this technique. And we've been able to treat some dense hardwoods as well. But despite these initial successes, there's still a tremendous amount of work that has to be done before this tool can reach the field. And I know that I'm not capable of getting it all the way there by myself. And even with a really strong team and plenty of resources, I know that it may still be too long before this tool reaches the field to save some incredible species of trees from extinction. And although I really do believe that the tools that we're developing will make a huge difference for timber and wildlife trafficking, I know that these efforts alone still won't be nearly enough to stop a mass extinction. But what I have realized in this transition that I've made from being trained as a biomedical engineer into becoming a conservation engineer is that conservationists are a lot like doctors for our planet. And in the same way that there are countless diseases that are faced by medicine, we have countless problems that are threatening the health of our planet. But as humans today, our lifespans are significantly longer than they were just a few generations ago. And this change has occurred because doctors started working in teams. They started working with chemists, with physicists, with engineers, for example, to develop new methods and new tools that could take on the toughest challenges facing medicine. And I believe that if we begin supporting conservationists the same way that we support doctors, that if we begin forming interdisciplinary teams around the toughest challenges facing nature, that similarly we can extend the survival of these incredible species and these incredible ecosystems that are essential for the health of our planet. Thank you.